Welcome to Season 6, Episode 2, Tiny Expeditions. This season is all about brain health. My name is Chris Powell. I'm your co-host for this episode.
And I'm Dr. Sarah Sharman, here to help you understand the science.
So, Dr. Sharman, what science are you helping us unpack today?
Today, we're going to dive into one of those buckets of brain diseases we talked about last week, which, if you missed it or you forgot...
No, they all listen. We checked.
Those three buckets were neurodevelopmental, which occur more early in life, neuropsychiatric, which are more middle life, and neurodegenerative, which occur later in life.
And the neurodegenerative is, we mentioned all of them, but we kind of camped out there last episode.
Exactly. So this time we're going to zoom in on neurodevelopmental, which, as their name suggests, occur when you're developing. So that means a lot of the symptoms show up in infancy or in young children. And unfortunately, a lot of them are actually pretty rare.
So rare disease inside of infants and children. To help us wrap our mind around, let's put ourselves in a hypothetical for a moment, right? Let's say that we're a parent, and a new parent at that, not like the fourth child, because the fourth child comes and you don't care, right? But let's say it's the first child, and you've waited all of this time and you've been expecting this child, and you're a bit nervous, right? Like there's a little bit of fear in that we're going to mess this child up. So you go into the hospital and you have this child placed in your arms, and it's like you fall in love instantly, and you want the best. You want everything to just go right for this child, right? And so we take the child home and we're doing the best we can, and we start comparing milestones, because that's one thing we can do. We'll go on the Googles, we'll start searching up on Google and seeing the milestones. And let's say our child starts falling behind in some of these. So we're not holding our head up at the right time. We're not yet being able to grab onto a finger. We're not able to, start pushing up these milestones. And we start to see the children around them passing them by. And if we don't have a lot of resources, first off, we may not even be aware that we're passing those milestones. But then we really find ourselves in a place of, where do we go from here? And even parents that have a lot of resources that can, see where these milestones are being missed and do have some idea of where to move forward, there's still these big questions of like, what do I do? I mean, where do we go from here? This is our child we waited for. Where do we go?
While we don't have answers to a lot of those questions, luckily there are a lot of people working on those, both finding out why these diseases happen and also increasing the access to diagnostics that can help provide these answers. So how about we talk to someone here at HudsonAlpha who's working on that type of thing?
Yes, my name is Susan Hyatt. I am a research faculty investigator here at HudsonAlpha, and I work on helping to diagnose people with rare disease.
And so when we talk about rare, I know the statistics might be hard, but what are we talking about when we say rare?
Rare for us is super duper duper rare. The statistics of, rare diseases affecting less than 200,000 people, but there are so many rare diseases. There are like maybe 7,000 at this point. And so to us, when we sequence individuals, a lot of times they are the only person in our study of 2,000 people that have a variant in that gene. So a lot of times we're talking about groups of people who have variants in a certain gene. There may be 10 or 15 that we know about, and that's about it. So super duper, never heard of it, rare.
This is super basic, but when it comes to these undiagnosed rare disease, what's at stake? Are we talking about just a risk of misdiagnosis, or what can the families gain from having a diagnosis? What's the important reason for the diagnosis?
Yeah, a lot of families really, it's just getting rid of that uncertainty of like, what is this? Part of it too is a lot of parental guilt, especially maternal guilt of, did I do this? Is this something that I did that caused their development to be different than other children? But a lot of it is trying to understand exactly what they have, how that may develop, what that may look like throughout their life, and to connect with other people who have that disease, who, of course, you're not going to find someone down the street probably that has that rare disease. And so trying to get them connected with other people. It also can give them an official diagnosis that then they can use to get other services as needed too.
I would assume for a lot of doctors, looking at genetic screening is probably not the first line of defense. So I would assume that this is a drawn out process.
Very much. So a lot of people, and it depends on who you see and what your insurance will pay for, unfortunately. But yeah, a lot of patients have that really long odyssey where maybe, let's do one of the biggest tests that will detect some of the more common rare diseases or some of the more common things. And then, okay, that's negative. Let's go to this next one, because there's usually that stair stepping, especially with insurance, and just in understanding, just in the frequency of it, right? So let's rule out the possibilities that we know about first. A lot of families go years just getting through all of those tests until they find a diagnosis.
So can you walk us through, you guys roll in this, so a kiddo is at the doctor and they finally order genome sequencing, and then that comes to you. What are you guys doing in the lab?
So, right. So in people who are part of our research studies, if they see like someone at Smith Family Clinic, the sample will come to HudsonAlpha. The DNA will get isolated from their blood, and then the sequencing will actually happen in the Genome Sequencing Center here at HudsonAlpha with Jane Grimwood and Jeremy Schmutz, and they will break the genome into big pieces, sequence those pieces, and then using a lot of bioinformatic tools, put those sort of back together to see where there are differences between that individual and the majority of other individuals in the world. And so then we will look at that data of where changes are happening and use a lot of different information put together to try and understand if this specific change has anything to do with a disease or what they're presenting with.
When we're talking about, especially diseases that affect the brain, right? And we bring in genomics into the picture, does that in some way help in talking about this? Does it in some way reduce the stigma a little bit?
Absolutely. Yeah, when people know that, yes, this is caused by a mutation that happened before you were born, that just happened to be in the wrong place, then there's clearly a genetic cause for this. Yeah.
So not only giving answers, but in a very real way, you're kind of giving people a new language to be able to talk about this and to be able to process this.
Right. Absolutely. Yeah.
So let's pause for just a moment here. Let's go back and remember our hypothetical. Actually, you know what? Let's get rid of the hypothetical because we've got plenty of real world examples at this point, right? Is there one that you can kind of point us to help us, land this plane?
Of course. So across the globe, 20 children went to their doctors with various neurodevelopmental symptoms. A lot of them had very similar symptoms, and their doctors couldn't figure out what was wrong with them. So that's 20 different kids in 20 different locations, all with similar symptoms, but nobody can figure out what's wrong with any of them.
And it's easy for us to say this, but this literally represents months and years of, agony of these families trying to figure out what's going on. And just to confirm, these are scattered all across the globe, and there's no way to figure out individually what they are, but it turns out that they're all actually showing the similar symptoms.
Right. So none of them were related. I don't think any of them really lived in the same city, and they're scattered across the globe. And it's not just 20 children and 20 families. I mean, it's probably a magnitude of doctors and geneticists all scratching their heads, trying their best to figure out what's wrong with these individual children.
So we need to figure out at this point, is there a way that all of these children are connected? And once we figure that out, is there a way that researchers can put the dots together to then return results to these families so that they can get the answers that they've so desperately needed?
So once you look at the genome and you find a variant that you think is interesting, how are you linking that to either the symptoms or the disease? And if you have an example that you can use to share, that would be good too.
A lot of the variants where we think it is interesting, it looks like a really bad variant, but no one has associated with disease yet. A lot of those genes we know that papers are still coming out establishing new disease genes. And so we want to kind of get ahead of that. Like, how can we find these variants even before those papers come out so that we can help with those papers? And so one of the things that we do is we reach out to the community, everyone else who's doing this, through a website called Gene Matcher. And we will submit a gene that we're interested in. And if someone else has also submitted that gene, we will get their email and we just email back and forth. Basically start a research collaboration on that gene to see if we can establish it as a new disease gene or if we have enough evidence for that. So one of those ones that we did recently is called ZMYM3, ZMYM3. And this one started, we found a variant in an individual who had some of those general features that we mentioned, just some delays and intellectual disability. And we found this rare variant in ZMYM3 that was, ZMYM3 is actually on the X chromosome. So the proband was a male and he inherited this variant from his mom. And so when you're on the X chromosome, it's even harder to kind of figure out whether or not something is pathogenic if you don't have family history and other things like that. But in light of all of the other genes that we find interesting, this one kind of fell into line with that one too, where it looked really damaging. And so we submitted ZMYM3 to Gene Matcher. And I should say too, before we submitted it to Gene Matcher, there was one paper where several brothers had a variant that looked damaging, ZMYM3. Again, there was sort of some overlap between those boys and this boy that we had sequenced, but it wasn't the exact same variant. And so we're like, well, that looks interesting, but we don't know. And so we submitted it to Gene Matcher. And over the course of several years, we collected information from all sorts of different clinicians all over the world. It was one of the biggest collaborations that we ever ended up dealing with I think 75 or 76 authors on our paper, which was amazing, but a lot. And I think we had around 20 boys who all had some similar features, all had variants that some of them were inherited from their mom on the X chromosome. Some of them were brand new. Some of them were de novo mutations, which we see a lot in these developmental delay syndromes. But the thing that we were still stuck with is, okay, this is on the X chromosome. Because we don't know, because we can't statistically show that these variants are enriched, how do we find out if they're really something or not? And so we ended up working with the Myers lab in order to do some functional validation, to take those variants, some of the variants, put them into cells and see what they did, see if they made a difference.
So Susan and this international team of researchers were able to pinpoint these variants in these kids' genomes that they thought were causing some of the issues. So that was all done computationally, looking at the genomes of these children, but they still wanted to confirm what was happening in the body, like what were these variants doing, what was it messing up. So that's where we go to another part of science, which is what we call the wet lab. So that's what you think when you're watching a movie and you see scientists in a lab doing experiments. So they brought in other collaborators that did some of that wet lab work. One of them just happened to be here at Hudson Alpha.
I feel like this is a great time for us to take a little field trip. Yes. Is that all right?
Yeah.
So let's go to the lab. And if you're listening to this podcast, you're still going to go to the lab with us. Don't worry, you're not going to miss out. However, if you watch this on YouTube, you can actually go with us visually. You can see what the lab looks like, and you can see all the visuals we're talking about. So just a little plug, go check out the podcast on YouTube, and you can see all the behind the scenes in the lab. But let's do it. Let's go to the Myers lab. Hey, Chris.
Morning.
Hey, Sarah. Hey, Chris.
How are you?
I'm doing well.
How's it going?
Another day in paradise.
Yeah. Well, thanks for letting us come into the lab today.
Mm-hmm. Absolutely. I'm happy to have you. I'm happy to talk about this project. It's one of my favorites.
Excellent.
Great.
So we've been talking with Susan Hyatt, and she has directed us to you.
So we really enjoyed our collaboration with them. We hope it leads to many more fruitful collaborations. This one was really fantastic and led to a really fantastic paper.
So why were you brought in on this project?
So the reason is the ZMYM3 gene that they identified in these clinical patients is actually, it codes for a class of proteins called transcription factors. And the Myers Lab has a long history with transcription factors. We really pioneered the way that you study the function of them, and the way transcription factors work is they have a role in recognizing particular places in the genome. They turn genes on or off in a very specific way, temporally, developmentally. And we suspected that the ZMEM3's transcription factor was going to be important in development, and that's why these clinical phenotypes happen, that mutations caused irregular development.
So once you're brought in on this project, how do you go about devising an experiment to explore this and to validate?
Right. So we did what we're good at doing, which we pioneered the method for looking at how transcription factors work. We knew in this case that we wanted to know how the ZMEM3 protein worked in its normal, it's what we call wild type configuration, the reference sequence, non-mutated. And we also wanted to see what would happen if we introduced the mutation. So what we did was you took a human cell culture line and we actually put in the mutation into that cell line's genome, the very specific mutations that were found by the clinicians. And then we did the assay that we always do, which is to see where in the genome does this protein bind. And we were able to do that for both the wild type and the mutant and saw some big differences. The way that we visualize how this experiment turns out, how the protein interacts with the genome, where it interacts, is generally through an image like this, where you have the genome lined up here in a linear fashion. We're zoomed into a very specific region of this chromosome. So we're tracked in pretty close. These are all genes shown here. So these tracks, these signal tracks here, show the strength or the amount of binding that the protein is showing. So the top two are the wild type, and you can see these top two have these higher peaks than these bottom two. This is the mutant protein and how it binds. Just by eyeball, you can see a reduction in binding here. You can see a massive reduction in binding here between the top two and the bottom two. And of course, we run computational analysis to actually give us statistical significance on the reduction of binding. But even by eyeball, it's striking.
And so no binding means no output of that gene?
This is the function of the protein, right? It has to interact with the genome in order to probably, in this case, affect the expression of this gene. So we hypothesized that a reduction in binding here would be a substantial change in expression of that gene.
So I assume there are probably a couple of different reasons why DNA wouldn't bind. Did y'all look into that at all?
Yeah, so the natural inclination here would be if there's reduced binding, it's because the transcription factor the mutation is giving it a reduction in the ability to bind. It actually turned out that was not the case for this particular mutation. And we had an inkling that there might be some other strange reason that the binding is reduced because of some annotations in at least one paper that might have said this particular part of the protein where the mutation isn't actually necessary to bind DNA. So we wanted to take it a step further and figure out how that mutation might have this particular functional impact. To understand this, you need to understand basically the concept of a cell is you have a nucleus, a compartment inside the cell. This, of course, is where the DNA is. A terrible sketch of DNA. The blueprint for all the proteins in the cell. In order to make the protein, what happens is a RNA molecule is made, a copy off the DNA. This RNA, messenger RNA, is then exported out of the nucleus to where the ribosomes are. Interacts with the ribosomes and the cytoplasm of the cell and makes the protein. Okay, a lot of proteins do their work out here in the cytoplasm, that's fine. But what about a protein that needs to bind to DNA? It's got to get back into the nucleus. And there are processes that will enable targeting of a protein to get into the nucleus. And it turns out that this mutation is interfering with that process, and that protein is just not getting back into the nucleus. And I can show you some images on that we took on the confocal microscope of that.
Yeah, we'd love to see.
Yeah, that'd be great. We'd love to see it.
Let's do it. So we're looking at, if you imagine the drawing that I just made, the cell wall here is not shown. This is the nucleus. So we use a particular stain called DAPI to stain the nucleus blue. This is just to visualize where the nucleus is. Okay? And then what we did was we took the protein, the ZMEM3 protein, and labeled it, so now it's going to show up red. Okay, so this is the exact same shot, but it's only showing where the ZMEM3 protein is. You can't see the rest of the cell here, but you can see that it's highly localized to the nucleus. This is the wild type. When you overlay those two images, almost all of the ZMEM3 protein is inside the nucleus. A little bit is out here on the edges of the nucleus, about to go in probably. When we looked at the mutant cells that we made, we saw something very striking. So you see the blue nucleus, you see the red where the ZMEM3 is, and you can almost see the edges of the cell here, because that protein is staying in the cytoplasm. It's not coming back into the nucleus to do its job, to recognize and bind to DNA. And I have one more where these are two cells next to each other with two nuclei. And you can see the red is just all throughout the cell and very little is getting into the nucleus.
So in the immediate, you all use this information to help diagnose several patients.
Right.
Is there other future applications of this discovery?
Yeah, you cannot minimize the effect of at least giving an answer to a family with a sick kid. And these rare variants often go without a diagnosis, and there aren't that many people who look at these. People throw a lot of money at big diseases, at common diseases, and they should. Alzheimer's, for instance, is going to affect everybody, so we should be studying it quite a lot. The type of research that we did here is very foundational. There's no immediate impact in terms of a therapeutic that comes out of this. But what this does is it lays the framework for a potential therapeutic. Because we know now where the wild type ZMEM3 should be binding and where the reduction in binding is for the mutants. So we know what the gene targets for ZMEM3 are. And we know that ZMEM3 is important because of the phenotypes. So down the road, as technology progresses, these genes that we've identified where the binding of ZMEM3 is, they would be the targets for therapies, if not ZMEM3 itself. This is why foundational research is so important. There are no companies that are going to fund this kind of work because it's so basic. But in order to get to the point where you can make a drug, or you have an idea, a hypothesis that a company could address, this foundational work has to be done. That's why places like NIH are so incredibly important for funding this kind of research.
So through the power of this massive collaboration, several of the children in this study received a diagnosis, which is impactful for their families because they have an answer to a question they've probably had for years and years and years. Now, there's not no hope for the ones that didn't get a diagnosis because researchers like those in the Cooper and Myers lab are still working towards finding more answers, finding more genes that are involved in these diseases, and just reanalyzing genomes, and also using new technologies like long-read sequencing that can hopefully give a larger picture of what's going on for kids like these.
Well, Squidward, what have we learned today?
let's take a moment, as we did last episode, and let's kind of distill down. Let's do a TLDR, right? We know everybody was listening. Everybody caught everything we were saying. Yeah.
Sure they were.
But let's take just a moment and let's see what stuck out to us. So, Sarah, was there anything that jumped out to you, something that we need to remember from this episode?
Yeah, I think for me, I always tend to go towards the more science topics. But for me in this episode specifically, the power of collaboration. I mean, we talked about it time and time again that there were, over like 50 collaborators on this project, which brought answers to, upwards of 20 people. So I think for me, that was really powerful.
And even within that collaboration, like you have 2 labs here, but those 2 labs represent very different ways of looking at the science.
Yeah, exactly.
But we have one computational, one in the lab, a wet lab. So I thought that was fascinating. One thing that stuck out to me is this idea of rare disease. And we say rare, we think it affects really no one, but rare disease is a lot more common than what we actually think. And just to pitch for the next episode, we're going to hear even more stories about rare disease as well. So those are coming, but it's not as uncommon as what you think.
Yeah, absolutely. And I think one other thing for me is that, traditional gene variants aren't the only genetic changes that can make big impacts on health and disease. Like Chris explained to us in the lab, transcription factors, which ZMEM was one of them, also are huge players in health and disease.
And can we just throw out a big shout out to all of those doing basic research as well?
Yes.
When it comes to these rare diseases, the things that don't make as much money, so they don't get looked at as much, but they're desperately needed for these families who are struggling with these diseases.
Yeah, 100%.
So with that, we'll say thank you for joining us for this episode 2 of season 6. We hope you learned a lot about your brain and the research being done in brain health, and we'll be back very soon with episode 3.
We will. Tiny Expeditions is a podcast about genetics, DNA, and inheritance from the HudsonAlpha Institute for Biotechnology. We're a nonprofit research institute in Huntsville, Alabama, with a unique mission.
We bring together scientists and companies to develop and apply genomic advances to make a better world. That includes everything from brain research to agriculture for a changing climate.
If you enjoyed this episode, swing by your favorite podcast app or YouTube and hit that subscribe button. While you're there, please leave us a review. We really want to know what you think about the podcast.
Thanks for joining us.